Okay, now we're going to talk on osmosis. Osmosis, remember, also is under passive transport. What is the difference between osmosis and diffusion? The key difference is going to be, when it comes to osmosis, through a membrane primarily permeable to those certain particles. Okay, let me kind of explain. In diffusion, all of the particles in diffusion have the same freedom of motion. All of the particles have the same freedom of motion. That's number one. Number two, remember in my definition, I say particles look at their own kind. Particles look at their own kind. Okay, but all the particles have total freedom of motion. In osmosis, that is not the case. It's kind of a prejudicial process in that you allow some particles to move and you hold back others. You allow some particles to move and you hold back others. So it still is a net movement of selected particles from a region of high to low, as I mentioned before, but through a membrane primarily permeable to those certain particles. So not all the particles can move. All right, so in my little demo I did here to show you, I'm saying that in this case, assume only the C particles can move and the others cannot. They are blocked. Only the C can move. Now remember, particles look at their own kind. Now because particles look at their own kind, they really don't care about the others that can't move. Each particle kind is looking at its own kind. So if some are blocked, that's of no concern to those that can move. So assume that the C particles can move and the others cannot. Then the C particles will move from high concentration to low, and not even think about the other particles. All right, now, this is what I want you to understand in that situation. In this drawing right here, here is a beaker. There's a membrane selectively permeable, allowing some particles to move and not. In this case, water can do it. In this case, now I'm gonna go back. As a matter of fact, let me kind of clarify something else. In a book, generally, you will see that osmosis is the net movement of water particles from a region of high concentration to low concentration of water particles through a membrane permeable to water. Why do I not have the water issue? Because osmosis came from chemistry, not biology. So osmosis is net movement of certain particles. That means it doesn't always have to be water if we go back to the root of it, which is chemistry. Why in a biology book do they say water? Because that is generally the case in biology, because everything in life is primarily water. So osmosis is not only dedicated to water, even though the biology books say that. But in this case, since this came from a biology book, we're saying that here's a beaker, here's some particles in here. Okay, here's some particles. Now, in this case right here, I want you to understand, this is a lower concentration of solute. So this is a solution. This is a lower concentration of solute, which means if there's not as many solute particles, there's more water particles. Let's look at that again. This is a solution. If there's not as many solute particles, there's more water particles. See the equal levels? This has more solute particles. That means there's less room, let's say, for the water. So the water would be in a higher concentration here and lower here. Higher concentration here, lower here. Since in this case, we're using water as the selectee. You're letting it move and you're not letting anything else move. That even though this membrane is permeable, look what happens. Water moves in this direction. Now check this out now. This membrane is permeable to water, freely permeable, but yet there's a higher amount of water on this side, even though the membrane is freely permeable. Even though the membrane is free, notice the higher level. Because the water is going to do its thing. Forget about the sugar particles. Each particle looks at its own kind. Here again, looking at it again. Okay. So when we come to osmosis, we're speaking of, in this case, a solution with a solvent and a solute. A solvent and a solute. Say it again, a solvent and a solute. Osmosis exerts a pressure. Osmosis exerts a pressure. It's called a colligative property in chemistry. Diffusion does not. Osmosis exerts a pressure. Diffusion does not. Why doesn't diffusion exert a pressure? Because all the particles can move equally. Osmosis exerts a pressure because it's a prejudicial process, and that selectee will exert a pressure over those that are not selected. So osmosis exerts a pressure, diffusion does not. The pressure exerted by osmosis is called osmotic pressure. The pressure exerted by osmosis is called osmotic pressure. The pressure exerted by osmosis is called osmotic pressure. Okay? So when we start talking about osmosis, and we talk about the solution, this is what I want you, these are terms I want you to start studying on. Hyperosmotic, hypoosmotic, and isoosmotic. Okay, let's go over here. The hyper, hypo, and iso, this is the way you have to think about it. A little tidbit. Always think about the solute amount. Always think about the solute amount. So if I use this for example, this side here has more solute particles. Therefore, it is hyperosmotic. Hyper. This side has less. This side has more. This side has less. The hyper has to do with the concentration of the solute. In order to have a hyper, you had to have a hypo. It's a relative thing. If both had the same amount, it's called isoosmotic. If both had the same amount, it's called isoosmotic. So hyper, hypo, if both had the same amount, iso. Now here's a little something I want you to think about. The hyper side always acts as a vacuum for water. The hyper side always acts as a vacuum for water, because the hyper side has more solute and less water. So water would come down its concentration gradient. The hyper side, that's a little tidbit for me, always acts as the vacuum for water. So let's go back to our terms. Hyper, more solid particles. Hypo, the lower side. Iso, the same. Now this is what you will see in clinical and general in biology. If the container, which is not this container here, because this is glass, if the container that the water is moving in and out of can stretch, if it can stretch, then we use the term from Latin tonus, to stretch. Tonus, to stretch. Now, when we're talking about life, we're talking about moving in and out of blood vessels or moving in and out of cells. Particularly moving in and out of cells. So, we use these terms, hypertonic, hypotonic, isotonic. They are synonymous to these, but you're dealing with an entity that can stretch like a cell. So to put it another way, the hypertonic side will act as a vacuum for water. If you have a hyper, you have to have a hypo. Iso of both sides. Now here we go with something. Now I want you to check this out. A red blood cell is isotonic in a 0.9% sodium chloride solution. A red blood cell is isotonic. That means if you put a red blood cell in a 0.9% sodium chloride solution, how do you make that? That would be 99.1 parts water and 0.9 salt. That's as simple as that. But if you put a red blood cell in a 0.9% sodium chloride solution, water will come in, but the same amount coming in will go out. The same amount, because at an iso, both sides are the same, so as much out will come in as it goes out. That's why 0.9% sodium chloride solution, quote, is called normal saline. That's why 0.9% sodium chloride solution is called normal saline. So Here would be a cell. We're looking at molarity here, 0.2. Here's the environment. So right out here, you have a 1, 2, 3, let's say. Here you have a 5. What's that going to mean? That this has more solute hyper than this. What did we say? The hyper side acts as a vacuum. So let's go to this. The hypertonic, I'm adding all of these up to that, and water gets drawn in. Now some comes out, remember, because it is a net movement, but more comes in than goes out. Then eventually these will reach equilibrium. Here's a red blood cell. Okay, we want to study on this. In this particular case, this is a hypotonic solution. That means the inside the red blood cell would be hypertonic. It acts as a vacuum. Then the water would come in. The water would come in and the cell may burst. Okay? We use an animal cell instead of plant. Same thing here though. It's just that you have a cell wall and this would make the plant stand up because this inside here would come in. Okay? Isotonic. As much comes in and goes out. That would be a 0.9% sodium chloride solution, speaking of this red blood cell. Here, hypertonic on the outside, greater than 0.9%. Water would come out. The cell would then shrivel, what we call crenate, and so forth. So here's another example that isotonic, as much comes in and goes out. Hypertonic solution. That means in this case, the solution, it acts as a vacuum. And here's hypotonic. Okay. So what you want to do is start thinking of molarity and molality and putting red blood cells in and so forth like that there. I wish this was face-to-face. I would do more. All right. Facilitated transport is a situation in which that you have to have a transmembrane that the particles still are moving down their own through their own energy. But the problem is that they don't have the best criteria. That means they generally would be water soluble at moderate size. Water soluble, moderate size. So they don't remember to go in and out. You need to be small, lipid soluble, uncharged. So these need a special door. Here's the key. These need a special door to help them in and out, even though they have their own energy. One good example of this is glucose. One example is glucose. Okay. Glucose is a fairly large molecule and it's water soluble. That means glucose is not going to want to go through the lipid layer. So what you have are these called GLUT transporters. You have these GLUT transporters. Here's some of the examples. Because glucose needs to enter your cells to get energy. Sugar needs to enter your cells to get energy. Now, these GLUT transporters, some of them need insulin. Some of them need insulin regulated. There are different kinds. Some don't, like in the brain. But these are transmembrane integral proteins. Let me go here. These are transmembrane integral proteins in order to let, let's say, glucose. It's not the only thing, but that's one example. Now that means then that the reason people have high blood sugar is because some of your cells require insulin to put the transporters in the cell membrane. Some of them do. Fat cells, skeletal muscle, and heart muscle cells need insulin. The others don't. In order to put these transmembranes. So if you don't, then the blood sugar will get high. If you don't, the blood sugar. I'm going to end this and